that'll be that'll help you. That we'll need to use in characterizing the first algorithm I show you, the concept of ultrametricity. So this is a property of the distance matrix. Remember what the distance matrix was? This was the pairwise distances between every pair of extant species, right? This was going to be the basis from which we construct the tree. You're somehow given the pairwise distances between. You're told that human and chimp are separated by X million years, or by X substitutions per nucleotide, something like that. Some measure of distance between this pair and this pair and this pair. So ultrametricity is a property of these distances. A given matrix of pairwise distances would have this property if for every triplet XYZ, two of the pairwise distances are equal and the third is less. So in this case, there will be three pairwise distances, right? Human chimp, human chicken, and chimp chicken. And if the human chimp, or let's say the human chicken, distance between human chicken is equal to distance between chimp and chicken, and both of these are greater than the distance between human and chimp, right? Two of them are equal, and the third one is less. That's what's shown here. If this holds true for every triplet of species, as per the distance matrix that you're given, right? The distance matrix that you're given, if it satisfies this property, you say that it's an ultrametric distance. Okay? And you can see the intuition here. If these branch lengths, if this is the true tree, right, and the path lengths are the true evolutionary divergences between these species, and the divergence is measured in time, right? You would expect this time to be equal to this time, so that these two distances are going to be equal, and this distance is going to be less, right? So that's intuition that this property tries to capture. So the other concept is that of a molecular clock hypothesis, or a molecular clock in general. This hypothesis is said to be true if the extant species S1 through Sn have evolved at the same rate of evolutionary change. Let's go back to this one, and in fact we can make something else here, yeast or something. Okay. If the branch lengths reflect time, you expect the tree to look like this, right? You expect that whatever the common ancestor of human and chimp was, lived some years ago, 5 million years ago maybe, and then since then, this lineage has diverged, has evolved for 5 million years, and so has this. Correct? So this branch length should be the same as this branch length, if these are time. Right? So that's obvious. But that does not mean that the evolutionary distance, okay, if instead of time, if you start looking at some other measures of evolutionary distance, what could be other measures of evolutionary distance other than time? I've mentioned some before. How would you measure evolutionary distance? Yes, Liam?
Like number of shared genes?
Number of shared genes, very good, yes. You look at the entire genomes, number of shared genes. If that's 100%, then distance is 0. Anyone with another suggestion? A quantitative measure of evolutionary distance. That's not time. Casey?
Edit distance?
Edit distance between what? Species meaning you can't edit a human into, like, you mean the genome?
I was just going on a base principle.
Between the genomes,
Yeah.
right? Yes, yes. So the number of differences between the two genomes, the number of substitutions. That's what edit distance would be. Any of those is fine. I mean, it's a reason. These are reasonable ways to measure the evolutionary distance between a pair of species. Okay? So if you're defining evolutionary distance as the number of substitutions, right, you can go ahead and actually calculate this. These genomes are available, you do the alignment, count the number of gaps and mismatches, and that's the evolutionary distance, and you can construct the matrix that way. If that was the definition of evolutionary distance, and the branch lengths are supposed to reflect that, you don't necessarily expect this picture that you expected with time. These two branches don't have to have the same length. Correct? Because maybe something happened in the evolutionary apparatus, and along this branch there are very, very few changes, while this branch has a higher rate of change. It's possible, and it's actually true. It does happen. So the molecular clock hypothesis says that that's not happening. It need not always be true. In some cases you may find it to be true, but the statement here is that each of these species here is at the same evolutionary distance from the common ancestor. So indeed this is equal to that in terms of the evolutionary distance that you are using. This is equal to that, this is equal to that. Not just in time, but whichever evolutionary distance you're using. If it's number of substitutions, it means that the substitutions have been accumulating at the same rate along every branch. Rate meaning rate with respect to time, and therefore the branch lengths are proportional to time, and therefore they have to have this property of being equal, for siblings. Right? A molecular clock tree is a tree that satisfies this hypothesis. Okay? If you say that the phylogeny of these 4 species is this, and these branch lengths are equal, this is equal to this, and so on, meaning you are saying that the mutations have accumulated at the same rate. It's your claim. You're making a claim based on when you draw this kind of phylogeny. And that would be a molecular clock tree. Okay? Because it's a tree for which the molecular clock hypothesis is true. And I've said that the alternative could look something like that. For some reason, if the human lineage was not mutating, if since the speciation event here, if the number of mutations has been very unusually small, well, small compared to, let's say, this, or this has undergone fast mutations, rapid mutation, then these two, even though in time these two are both 5 million years after this point, in terms of the evolutionary distance you are using, that's the number of substitutions, they would be different. So this is the alternative. For this, the molecular clock is not true. Now, yes, Liam.
Is there another alternative for the scientists that reject evolution because of their religious beliefs?
Is there another alternative of what?
These ideas. Like if you don't believe in evolution.
That's a rhetorical question. I don't think I can answer that. Yeah, all of these concepts are conditioned on you assuming. Well, it's a model of data. You could think of it that way, right? Whether, I mean, your definition of truth can, I don't want to get into a philosophical discussion here, but you understand that this is a model of data, right?
It could be you could define the distance as the amount of time between when God made one species and made the next species.
Yeah, sure, up to you, yeah. It is a model of data, and then interpretation of that is up to people, however they want to do it. Okay, so here's the relationship between the two concepts I introduced, that of molecular clock and ultrametricity, which is misspelled here. In a molecular clock tree, all triplets are ultrametric. And that's kind of obvious from here, right? The way I introduced a molecular clock tree, take any triplet, this, and this, right? This will be equal to this. Well, sorry. This will be... Sorry, no. Here the distances are not from any internal. Distances are pairwise distances. So distance between this and this will be equal to distance between this and this. And the distance between this and this will be less. That's the ultrametric property. And that's going to be true no matter which three species you pick here. The other way is also true in a sense. If all triplets in your distance matrix follow that property, so if your distance matrix is ultrametric, then there is a uniquely defined rooted molecular clock tree for that distance matrix. So you're given a matrix of pairwise distances, and you check that for every triplet, this property holds. The two of the distances are equal and the third is less. Then you can be assured that there is a tree you can output which will have this nice property that every pairwise distance will be equal to the corresponding pairwise distance in the given matrix. And that was how I motivated the problem, right? You're given the distances and you have to find a tree that matches those distances. Matches in the sense of path lengths between species being equal to the pairwise distances. Is this clear? Okay. But otherwise, it need not. I mean, if your distance, I cannot guarantee that any given pairwise distance matrix will result in a molecular clock tree. If it's not ultrametric, it won't. Okay. So, in a distance-based method for phylogeny reconstruction, you're given by the n by n distance matrix Dij, and you want to come up with a tree that best fits the distance matrix Dij. In other words, you find a tree T such that Dij of T, this is just the path length between I and j in the tree. Okay? Is equal to the corresponding pairwise distance for every pair. And such a tree exists if the given matrix was ultrametric. I've already stated that. So let's look at one algorithm that will do something like this, that will produce a tree. Okay? And we've actually seen the algorithm in a different guise earlier on. Anyone remembers something that looked like a tree construction from points? Hierarchical clustering, yes. Where we said that every given point is its own cluster, and then we started grouping closest pairs of clusters and saying that those pairs, that pair of clusters defines siblings of a common parent, right? It's exactly the same algorithm, except now we'll put. Someone had asked this the other day. Why aren't we putting edge lengths on that tree that we were constructing? And that's all we are doing now. We are going to add edge lengths to the tree that we construct. Otherwise, it's the same algorithm. Okay, there the goal was just to cluster, not to build a tree. The goal is different here. The same algorithm is going to be used. It's called UPGMA, and I forget. So here's the full form: unweighted pair group method with arithmetic mean. So what is it? It's the hierarchical clustering algorithm we saw, with the distance between clusters being defined as the average pairwise distance. You're all fresh off from homework 4, where you did this, except that the distance matrix you probably, I don't remember what detail, maybe I said it should be the minimum distance. Was that what I asked? But I had mentioned in class that it doesn't have to be the distance between two clusters can be defined in whatever way you want. For example, it could be the pairwise, so average pairwise distance. Take 1 point from here, 1 point from here, compute distance, do this for every pair of points, take the average. So that's what the distance here is defined as. Okay? It outputs a tree whose leaves are extant species, so it's a phylogeny. And it allows perfect reconstruction of a tree if the distance matrix is ultrametric. So if the given distance matrix has the ultrametricity property, then what this outputs will be the right tree. It will be the exact match to the distance matrix. Okay? Which I had promised exists, based on the previous slide statements, and that's the tree that this will find. But it'll work for any distance matrix, ultrametric or not. It'll do something, right? It's just that if the matrix is not ultrametric, you're not the output will not exactly match the distance matrix. But it'll produce something. Okay? And I don't have a better way to characterize what that something will be, apart from just describing the algorithm. So, to repeat myself, given the matrix, this method will find a tree. If the matrix was ultrametric, this tree will be a perfect match to the matrix. If it was not, this will be some tree. Okay? To repeat myself, the distance between clusters in this hierarchical clustering algorithm will be the average pairwise distance. This is the complicated way of saying that. Here's the algorithm. And you should really understand the steps of this algorithm. Ideal for asking exam questions for them. So initially, like the hierarchical clustering method, each point, each xi, each extant species is its own cluster, ci. Okay, and you're also constructing a tree, so each of these is placed at height. I'm going to define heights along this dimension. That's at height 0. Find two clusters such that Ci and Cj such that Dij is minimum. So that amounts to, at this step, finding the closest pair of points. Right? So find the closest pair of points and let those be Ci and Cj. Then you define CK as the union of CI and CJ. So far, exactly the same as the hierarchical clustering. So this is in the clustering method, you'll just make this a cluster. But now you'll do something more because you're not interested in clustering, you're interested in drawing a tree. And so you add a vertex to this tree. You introduce an internal node in the tree for CK. Okay, so you introduce a node for CK, and you place it at height dij by 2. Okay, so this height of CK is going to be dij by 2. Which kind of makes sense here, right? Because the distance between these two is dij. And I told you we are going to output a molecular clock tree. So the height should be dij by 2. So, it'll be a tree that goes like that. The length of each edge, you'll now put lengths on these edges, and in this case it's trivial. The length of edge between CI and CK, the length of this edge, let me draw it a slightly different way. The length of this edge should be the height of CK minus the height of CI. In this case, the height of CK is dij by 2, the height of CI is 0. So the length of this is dij by 2, and the length of this similarly is dij by 2, right? And then you can delete the clusters CI and CJ. You can forget about them now. So now there are 1, 2, 3, 4, and 5 units to work with, right? And again, just like you did in the homework, you'll compute pairwise distances now with no row and no column for CI and CJ, but with a row and column for CK. Pairwise distances will be computed as per the definition, the average pairwise distance, right? And so you'll have a new distance matrix. You'll find the closest pair of clusters, and that, let's say, that ends up being this and this. Okay? Then what will you do next? That'll be you'll introduce a new internal node when you're trying to merge this cluster and this cluster. Correct? Then you'll define the height as the distance between this cluster and that cluster divided by 2. Let's call this, A. So this will be DAK, distance between cluster A and cluster K, divided by 2. Correct? What's that going to be? What's DAK? DAK is defined as the average pairwise distance between a point here and a point here. Well, the points in this are these 2 points. So DAK is going to be DAI plus DAJ by 2. Right? That's the pairwise distance. And in fact, if this was an ultrametric matrix, these are going to be the same as well. Okay? So I guess by now you understand, what happens in every iteration. You put a point at this edge, then you draw edges like this, you put lengths on this as per these rules, and move on. It's really quite simple. Just go back and try this out with an example, and you'll understand what's going on. And in the end, you would have built a full tree. Only one cluster remains, and that's the tree that's output. Here's an example. So that's the UPGMA algorithm. Okay, very simple. It produces a molecular clock tree. So what it produces will always have this property of things being at a certain height. I mean, the path from the common ancestor to every leaf will be the same length. And you can almost see that here. Right? There's no way to get trees like that with different, it just won't produce such trees. Okay? And so if the molecular clock is really not true for the species you're looking at or for the data you're looking at, sometimes for the same set of species, if you base the tree on certain genes, the tree will look one way. If you base it on some other genes, it will look different. And so in that case, if for the genes that are guiding your reconstruction, that define the distance matrix, if the molecular clock is not true for those genes, then the tree output here will not agree with your data. Did you understand that? It's kind of non-intuitive from. I've been saying that our goal is to build a phylogeny for species, right? And the phylogeny, I've been saying, need not have lengths representing time. It could be rate of evolution. But rate of evolution is not homogeneous across the genome. Maybe in one part of the genome, in each of those species, the rate of evolution is the same. Maybe in another part, parts for genes related to brain function, the human lineage has been accumulating mutations at a faster rate. Right? So the phylogeny actually could be different depending on what data you're using to build that phylogeny. In which case it no longer remains just a relationship between species, it becomes a relationship between the considered portions of those genomes. Right? And so if the data was taken from a gene related to brain function, and for that gene, the rate of evolution has been faster along human line lineage compared to other species, then the molecular clock is not true for the data. But the UPGMA cannot do anything about it. It'll always say human and chimp, like that, same length. Okay? So that's the major pitfall of the method. People sometimes do want methods that reflect distances more accurately.
So this also wouldn't work if you had DNA from extinct species, because they've stopped evolving, obviously.
You want to place the extinct species onto this? Is that?
Well, I was saying that it would also fail if you tried to do that. Correct? It would produce a tree that would say that this extinct species is still alive today. That would be the
Right. Yes. Right. If it would treat every given species as an extant species. Yeah. And so you're correct. This is just an example of a case where the correct tree is not a molecular clock tree. The distance matrix is not ultrametric, but if you computed the pairwise distances, built a distance matrix, and gave it to UPGMA, it might produce something like that. So as I've mentioned, the output tree does not necessarily match the distance matrix, except in the case of an ultrametric distance matrix, in which case it'll find the molecular clock tree corresponding to that matrix. Okay, so that's one kind of approach to building phylogenies based on distance matrices. Next, we'll look at a rather different approach, which doesn't even look at which doesn't really look at pairwise distance matrices. It looks at the underlying alignments that produced that distance matrix. So what do I mean by that? You have and that's called a character-based tree reconstruction. So again, you're given human, chimp, and chicken genomes. And instead of taking each pair and computing their pairwise distance, what you can do is to just align those genomes with each other. Okay? And in fact, you would have to do that anyways if you wanted to construct pairwise distances. Right? That's how you would have computed pairwise distances, by taking every aligned pair and counting mismatches or counting gaps and so on. But here you don't do that. You actually just use the alignment itself to construct the tree. That's the distinguishing feature of this approach. So now what you have is you've aligned these. For simplicity, let's assume that there's no gaps, or that we've gotten rid of all the gaps. Okay? So that what we have looks maybe like this. Okay, it's an alignment with some mismatches in some columns in some species. This is human, this is chimp, and this is chicken. Okay? So what you do is write down those sequences. Add the leaves. Okay? This is how you start. This is how you write down the input itself. And then your goal is to build a tree, which maybe it would look like that, with sequence labels for the internal nodes as well now. So it could be TACGT at this, and TACGT here as well. Okay, something. You have to. It's not known what these internal node sequences should be, because you don't have those. But your goal is to find a tree and to assign labels to these internal nodes. Okay? This is another way of phylogeny reconstruction, because you are trying to reconstruct the entire evolutionary history. You're trying to guess what really happened with time. For example, you're trying to guess that this common ancestor was TACGT, and that sort of makes sense because that these two are identical. Why would it be something different and both of them will change in the same way? Why that? Rather not have any change. And you're trying to guess that this was also TACGT, so that there's no mutations here, and there's a couple of mutations here. An A to a C and a C to G. So you're guessing a tree with labels for the internal nodes, the sequence labels, the internal nodes, so that you know exactly what mutations happened on each branch. Okay? And the guiding principle, I haven't said, what should be your objective function or your guiding principle for guessing these labels. Let's look at the second bullet. You make any guess, that will define the mutations that are happening on that branch. Right? I just told you that if this is your guess, this has no mutations, this has no mutations, this has no mutations, this has 2. The number of mutations is also called the Hamming distance between those sequences. Right? Remember, there's no gaps allowed. So this is the Hamming distance. And so you note down the Hamming distance on every edge. And the sum of these Hamming distances is defined as the parsimony score. And the reason for this name is that you will try to find the guess, the tree, and its labeling for which the parsimony score is minimum. So you're trying to be parsimonious with respect to mutations. You're trying to find a tree and its internal node labels that can explain all the extant sequences with as few mutations as needed. That's why parsimonious. Okay? In fact, when you say that 2 mutations happened here, that's already a parsimonious guess. You could have had a G change and then change back to a G. But you're not counting 4 mutations, you're counting 2. You're assuming the simplest explanation in terms of evolutionary events for what happened from here to here. And when you sum these things up, you're assuming the simplest explanation of what happened over the entire tree, given these guesses. Maybe by changing the guesses, maybe the number of mutations will change. Maybe you could come up with a smaller parsimony score. And so the goal is to find the tree and its sequence labels so that the parsimony score is minimized. So that's the Occam's razor principle, the simplest explanation for the data. And I should mention that here the sequences we are dealing with are nucleotide sequences. But the idea of taking the number of changes is fairly general. You can have other things as well, on other ways to describe these species as well. Right? Number of eyes or legs or shape of a beak or fin, and as long as you have some sort of a distance value for this character changing to that character. Here, a C changing to a G means 1 mutation. Okay? You could say that a beak changing to a longer beak is, has score of 10. It could be anything, as long as you have defined distance between those states. All right, so what we are going to do with this kind of data is, instead of computing pairwise distances and and doing UPGMA, we are going to put those sequences at the roots, at the leaves, and we're going to guess a tree, and we're going to guess the internal sequences, the internal node sequences, so that the number of mutations is minimized. That's our guiding principle now. Okay. So here's a couple of examples. It's really, unfortunately, data. So it's not the best example, but these are 2 different trees for 2 different data sets. In this case, you can, in fact, these are 2 different guesses.
Looks like the same sequences, but just arranged differently.
Really, they are? Oh, good. That's good. Yes, yes, yes. Oh, this one's here, right? That's right. So it's the same sequence, the same data, but two different guesses for the evolutionary history. Okay? By a guess, we mean, how, what's the topology of the tree relating them, and what are the internal node sequences. And once you've guessed what happened at the internal nodes, you can easily count the number of mutations, 2, 2, 1, 1. So that's 6 here, and there's 5 here. So this is more parsimonious, fewer mutations. You would prefer that guess over this guess. Okay? So that's our goal, to find the tree with the lowest parsimony score. And this is a very daunting task. But I'll simplify it for you and then ask you for ideas. I'll define the so-called small parsimony problem. Small for easier, actually. Where the topology is given to you. So you're told, this is the topology. This is what I mean by topology. This was the data. You're additionally told that this is the topology of that these two are siblings, and this is this is next, further away. And you have to guess the internal node sequences. Okay. So you're given a tree T with each leaf labeled by an M character string, in this case 5 character string, labeling of the internal vertices of this tree, minimizing the parsimony score. That's the small parsimony problem. Okay? And in fact, I'll give you a further hint towards solving this problem. Which is that because we decided not to worry about gaps, we can assume that this T corresponds to this T corresponds to this T evolutionarily. Okay? This A, this C correspond to each other evolutionarily. Which means we can solve the problem on this instance. Try to guess what was here, and then separately solve the problem for this instance. Try to guess what was here, and then put together those guesses one after the other, and that's the right answer. Okay? We don't have to worry about the second position in this common ancestor corresponding to this because there was an insertion or deletion. No. Okay? So I really simplified the problem. Once you can solve this, whatever time complexity it is just gets multiplied by the length of the sequence. So that's a manageable thing. It's linear in the length of the sequence, provided you can handle this efficiently. Okay, so I've made the problem substantially simpler, but it's still difficult. Because you basically so the naive approach or the brute force approach would be to try all 4 possibilities here, and if the tree is bigger, all 4 possibilities at every internal node. So you'll have to try that many combinations of guesses for the single character at each internal node and score that guess, that complete guess, and then pick the best one among them. The number of internal nodes is about the same as the number of leaves. So your algorithm is exponential in the number of leaves. Not good. Okay? And so you'd immediately say, well, that's kind of stupid. I can be smarter. And what you can do is, look, I have a t here and a t here. I'm going to place a t here for sure. Okay? But you can't, you can show that this is not, in general, a good strategy. This is the greedy strategy, right? Bottom-up, greedy allocation of the character, because the cost of making this guess is related to what's on the other side as well, right? What's on the other side of this node, the remaining part of the tree. So I won't go into explaining why the greedy is not okay, but it's not okay. So we're stuck with this method, which is not good. And so that's the meat of the algorithm I'm going to show you. And our mantra is, when in trouble, use, in bioinformatics, when in trouble, use. There's only one smart algorithm I've ever presented in this course. Dynamic programming, okay? So there's going to be a dynamic programming method to solve this in time linear in the number of species, as opposed to exponential in the number of species. So that's what we'll do. Before moving on, we'll just generalize the problem a little bit by defining the so-called weighted small parsimony problem. It's a more general version of what I described, where you have a scoring matrix. Well, you have for every pair of characters, just like in the alignment situation. It's not just match or mismatch. It's not just mutation or non-mutation. It's not just 0, 0, and and 2. The A to a C could be a different, could have a score of 3. The C to a G could have a score of 1.5. Okay. So a generalization of the 0, 1 match mismatch scores to something else if you want to be more realistic. For the same reasons that we did use that in alignment. In alignment, we went from match mismatch counting to scoring matrices because we know that certain pairs of nucleotides are more similar than other pairs of nucleotides. Right. For small parsimony problem, the scoring matrix is simply the Hamming distance, the match mismatch between characters. But the weighted parsimony problem, the scoring matrix could look like that, instead of the small parsimony problem where it was always 0, 1. Okay? So you're given a tree T with each leaf labeled by elements of a letter K, of a K-letter alphabet, and a scoring matrix. And then labeling of internal vertices of tree T. And here's the dynamic programming algorithm. Actually, I have an example. The key thing from this slide is the definition of the dynamic programming table. The first step in dynamic programming is to define what is the variable for which you'll compute recurrences. Right? That's the dynamic programming table. What do elements in your table represent? That's the first thing you define in dynamic programming, right? Then you define how to compute each cell based on other cells. That's the recurrence part. But the first part is the semantics of the table. And that's what this is shown here. ST of V. So there's going to be, the table is going to be distributed over the tree. There's going to be a table here, a table here, a table here. So these are the cells that you'll compute. All right? So there's going to be one table for every internal node, V. In fact, I think in the algorithm we'll put such a table for the leaves as well. So there's going to be a table for every node V. So that's the V part here. Okay? And at each V, the table will have elements for each character. So that's the T part. So the T, these would be the nodes in the table, in the tree. And these would be the characters in your alphabet. Okay? And so in this case, the T would be A, C, G, or T. Now, the number, the value of this variable is going to be the lowest parsimony score of the subtree rooted at this node if we placed a C at this root. Okay? So it has the flavor of the dynamic programming methods you've seen earlier. It's the best solutions value for a subproblem. The subproblem here is the subtree. Okay? And furthermore, it's the best solutions value or the subproblem under a certain local decision. The local decision here is the placement of a C here. If we placed a C here, what's the best score we can get from the entire subtree? If we place a C here, what's the best score we can get from the whole tree? If we place a G here, what's the best score we can get for this subtree, and so on. So that's the semantics, the definition of the dynamic programming table. The recurrence is given here for your reference, but we'll just look at it with an example. And so note that if we can compute these things in an efficient way, then at the end, all we have to do is take the lowest value among these four, right? Because this number tells me what would be the best score for the whole tree if we had an A here, if we had a C here, if we had a G here, if we had a T here. It has to be one of those four. Take the lowest value, and that's your answer. So you can see that if we solve this problem, if we fill in this table, this distributed table, we'll know our answer. Okay? So that's one thing to note. The other thing to note is what's the time complexity going to be, approximately? We've not seen the recurrence yet, so you don't know the answer, but there's 4 values to compute for every node. The number of nodes is linear in the number, is about the number of leaves, right? If you're only counting internal nodes, it's about the number of leaves. If you're counting all nodes, it's about twice the number of leaves. Okay, so it's about the number of leaves. So the total number of cells to compute is the total number of leaves times 4. Big O of that. Right? So as long as you can compute each of these numbers in some small time, you are going to solve the problem in time that's linear in the number of species, as opposed to exponential that our naive method was. So here's the example. You begin at the leaves. And here's the distributed table that we're going to fill in. For the leaves, the rules are very simple. So you're given the character at the leaves. And you're given the scoring matrix. I mean, in this case, intuitively, if it was all zeros and ones, if you were only counting matches and mismatches, then you can almost guess what, there's nothing smart you can do. I mean, this will be an A or a C, this will be a T or a G, and this will be either A or a C or a G or a T, and all of them will give the same score overall. So it's kind of it's a bad example in that sense. But the scoring matrix will change the situation for us. So we'll see that in the next slide. First, we're talking about the leaves. So for the leaves, if this is an A, that entry is going to be 0, because the best parsimony score for the subtree rooted at this point is, if there's an A here, is 0. And if there's a T here, then it's you put an infinity there, because you cannot put a T there. You have to have an A at that point. Right? So that's kind of the base condition. Infinity for everything other than the actual character at that leaf. Now, based on these two tables, you'll fill in this table. Okay? And to fill in, let's say, this entry, I don't remember which one. So that's this table. This is the scoring matrix, because this is a generalized problem, not the 0, 1 scoring matrix. This is the scoring matrix. So now you have to, and this is the recurrence that was on the earlier slide, which I didn't describe. Now it's the same recurrence. Okay? It's really, again, quite simple. If you want to put so this is our V. All right, this is our V. SAV is the number we'll put in here. By definition, it's the best score for this subtree if you put an A here. Well, that's easy to calculate. If you put an A here, what's going to happen here? Well, you could have chosen from you have 4 options for this node. One of them is 0, the others are infinity, so you have to choose 0. You have to choose an A here, in which case if it's an A and you're putting an A here as well, an A to A is, what did I do? It's not right.
Yeah, that's right. 0. Wouldn't cost anything.
No, but why is this 3? That's wrong.
The best score is going to be if you choose T, because it has.
Oh yes. Oh, okay. Thank you. No. I think ignore these edge lengths. Okay.
Yeah, that's right. Because if you choose A, then the cost to go to C is 9, right? And so that's worse than choosing T.
Right. But no, here I was discussing what happens when you place an A, and this is not relevant to that. This is relevant to the correct answer finally. If you put an A here, then you're going to pay 0 for this part. You're going to pay infinity for this part. No. An A to C, sorry. You pay how much? A to C is 9. You pay 9 for this part. And 0 for this part. So the sum is, so you're taking the minimum of both children separately and adding them up. Okay? So in fact, what you'd fill in here is 9. If you put a T here, then the minimum for this part is going to be T to A is how much? 3. That's what's shown here. And T to C is 4. So 3 plus 4, and then you'll put a 7 there. Okay? And so on. What's going on? Okay, this is just explaining what I just already explained. Already explained. So you put a 9 there. If you put a T, you'll get a 7 there. If you put a G, you get an 8, and so you filled in those numbers using this recurrence. Okay, it was kind of easier to calculate. For this case, because you really had to look at only one option. The others were infinity. But in general, you look at all 4 options that will be on the table. And you'll take the number there and add the distance to that, for that mutation. The score for that mutation. So maybe there is an example for, here's that example. We have 9, 7, 8, 9 already filled in. Repeat for right subtree, and we fill in 7, 2, 2, 8. Okay? You can just do it. And now we'll look at what happens for the root. And here you'll have to, if you want to place an A here, then you'll have to consider all 4 options for this point. If you also place an A here, then you get 9 plus 0. If you place a T here, then 7 plus whatever is the distance between A and T, and so on. So you have 4 options, take the min of those 4 options. Similarly, min of 4 options there, and ultimately you add them up, you get 14. Okay? So at each step, you'll have 4 options of what to choose for the child, given a certain parent. Among all those 4 options, you will add this number here to the distance from A to that option. Take the min among the 4 options, and that's what you put. That's the contribution from this part. Do the same here, add them up, you get the number there. Okay? You can go home and try this out, and you understand what's happening. So, and finally, you just take the minimum at the root, which is 9, and so you place a T there. So this is the trace back. And it's quite simple. You just take the best and the 9, and then you decide how the 9 came about, and you'd probably realize it came about by taking T here and a T there. Okay? This is called the Sankoff algorithm. And this slide just describes how you do the trace back and go down the tree and just fill in each to actually make the choice, the optimal choice for each node. Okay? So that's, and as I had already said, the total number of entries you're computing is linear in the number of species. And to compute each of them, you do a minimum over 4 numbers and a minimum over 4 numbers. And so it's all constant time. And the algorithm is linear. Okay? And you do this for every single nucleotide in the original alignment, every single position in the original alignment. And so it's the number of species for each position times the total length of the sequences. That's the time complexity. So, of course, I had simplified the problem for you by defining the small parsimony problem, meaning that the topology was given to you. Remember, all this was conditioned that you know on that you know the topology. But in general, you don't know the topology. I mean, in UPGMA, you were actually finding the topology, right? So the large parsimony problem is the full problem. To find the topology, and for that topology, solve the small parsimony problem, and then do this for every possible topology and find the best topology and the best labeling of that topology. right? So at least now, given a topology, we can evaluate what's the best labeling, what's the best parsimony score for that topology. And in principle, you could enumerate every topology, solve this problem in linear time, and finally pick the best topology. But there's a large number of topologies. We discussed this earlier in the very beginning of this PowerPoint. The number of trees, rooted trees, is very large. The problem is NP-complete. An exhaustive search would be possible only with a very small number of species. If you're an evolutionary biologist wanting to build a tree out of 100 bacterial genomes, it's out of the question. Okay. And so heuristics are used to heuristics meaning just things that sound like smart ideas, but you can't really say anything. They don't guarantee anything. Those are called heuristics. Heuristics are used to search this large space of topologies, to find good topologies, evaluate them using the Sankoff algorithm, and then pick the best topology. And I won't describe any details about these kinds of heuristics. I'll describe something on a high level. It's a branch swapping algorithm. It only evaluates a subset of all possible topologies, all possible trees. What it does is it has a certain topology in hand, right? A topology is a tree relating all the species without any labeling from the nodes, without any branch lengths or anything, just a tree. And it has a score for that tree. And then it does a small change to that tree to move to a slightly different tree, okay, a slightly different candidate topology, which it then evaluates. And if it's better, then it goes there. And then again, from there, it makes a small change to another tree, evaluates that. If it's better, it goes there, and so on. So it's a greedy algorithm. So what is this small change that it will do on a tree? It defines the neighbor of a tree. It defines a small change as one that's reachable by a nearest neighbor interchange. And that's the last thing I'll show you what that is. Let's say this is the current topology. And these are not 4 nodes. These are, the tree can have many nodes here. It's just that we're focusing on this branch. It's a certain view of the tree. And this branch, for this node, these 2 will be its children. And these 2 can have their own subtrees. In general, will have their own subtrees. Right? As I said, these are not leaves necessarily. These are internal nodes. So if you focus on any internal branch, in general, the tree will look like this. The branch will connect 2 nodes. Each node will have these 2 children. Maybe the root was somewhere here. Or it doesn't have to be. This could be the root, in which case, if this is the root, this is a child, this is its child, and there's another child of that. And then this has 2 children. So if you focus on any branch, in general, it'll look like this, where these triangles are further subtrees of these nodes. Right? So you focus on an arbitrary branch, and then you swap this part with that part. That's a different topology. So you see, you had A, B as siblings, C, D as siblings. You take this entire subtree from there and put it here, and the entire subtree from here and put it there. Right? You just cut, as if you've cut at this branch and you just stuck it there. This is what is a nearest neighbor interchange. And this has fundamentally changed the topology. Right? If you had replaced these 2, it would not be a fundamental change in topology. It's the same topology, just looked, seen slightly differently. But this is a fundamental change in the topology. right? So this is what it does. It chooses an arbitrary branch and does a nearest neighbor interchange. And it could have chosen this branch as well and would have done the nearest neighbor interchange after focusing on that. So there's many possible neighbors that it could evaluate. So I don't know what exactly it does. It could just try all those neighbors and pick the best one. What I've defined is, one neighbor one thing in the neighborhood of a given tree. There's in fact many things in the neighborhood, in the immediate neighborhood of a given tree, depending on which branch you choose to focus on. You could evaluate every neighbor, do the Sankoff thing on that neighbor, pick the best among the neighbors and go there and iterate. That's a heuristic. By the way, this is also a fundamentally different topology from, right, when we have, I said you exchange B with C, that's a fundamental different topology. You could also exchange B with D, bring D here and bring this there. And that's also a neighbor, right? So in fact, two neighbors if you focus on that branch. Right, so in fact, the greedy algorithm is that start with a tree T, move to a neighbor with the best parsimony score. So evaluate all neighbors, evaluate their best parsimony scores, and take the best among those, move to that. And that's it.